The overall goal of this method is to enable depletion-rescue experiments to be performed under conditions that preserve cellular integrity and protein homeostasis for functional analyses of biological processes at the single-cell level.
This method can help answer key questions regarding protein functions and structural requirements in cell dynamics, such as mitosis and cell differentiation.
The main advantage of this technique is to deplete a protein of interest and reintroduce at the same time variants of this protein at near physiological levels in a majority of a cell population.
Begin by coating glass-bottom dishes with fibronectin. To do this, dilute a 1 milligram per milliliter fibronectin stock 1:100 with sterile PBS. Add 1 milliliter to each 35 milliliter dish to cover the whole surface. Incubate for 1 hour at 37 degrees Celsius and 5% carbon dioxide. During the incubation, pre-warm alpha MEM supplemented with 10% FBS and an aliquot of 0.05% trypsin EDTA to 37 degrees Celsius. After 45 minutes of incubation, start preparing the cell solution. Aspirate the medium from a 10 centimeter plate of HeLa RFP H2B cells and gently rinse twice with 1.5 milliliters of 0.05% trypsin EDTA. Leave 0.5 milliliters of